Hey everyone, this is Dylan here, and today I'm going to go over the Kinyoun stain. So this is a stain that you use for Acid Fast Bacilli, or AFB. And the way you do the Kinyoun stain is that you use Carbolfuchsin as your primary stain for 4 minutes, and then you use your acid alcohol decolourizer just for a few seconds, and then you use your counter stain of Methylene Blue for 30 seconds. And so when you're done with that, what it means is that your AFB positive or Kinyoun positive organism will stain pink or red, and if it's AFB or Kinyoun negative, then it's going to stain the counter stain color, which is blue. So notice that that's the opposite of a gram stain pretty much. So just keep that in mind when you're reading the stain. And also note that your AFB organisms will stain gram positive as well. So that's another thing to keep in mind. And the whole principle behind this stain is that your AFB organisms will have mycolic acid in their cell wall. And what that does is that will resist the acid alcohol decolourizer. So what that means is that it will retain the primary stain color, which is that pink or red color. And your non-AFB will get decolorized from the acid alcohol, and then they will turn out the counter stain color, which is blue. So your AFB organisms traditionally include Mycobacterium, so that includes your TB or tuberculosis and non-TB organisms. But in 2018 there was a delineation between some of the Mycobacterium group. So what that means is that there's a new genus called Mycolicibacterium, and that includes some of your old Mycobacterium bugs such as smegmatis would be one of them. But that doesn't really affect the staining because those two genuses will still stain AFB positive, but there was a name change. That's something I just found out recently, but it did happen in 2018. So those are going to be your bugs that are going to stain AFB positive, and not to get confused with some of the bugs that will stain partially acid fast. And what that means is that for the modified Kinyoun stain, the following bugs will stain positive, but for the AFB stain, they will stain negative. So these bugs include Nocardia, Dietzia, Tsukamurella, Rhodococcus, for example. Those are all partially acid fast, but they should not traditionally stain positive for your true Kinyoun stain. So when would you do the Kinyoun stain? So you would do this stain on any suspected AFB organism from the gram stain. So remember, for the gram stain, your AFB organisms are going to be gram positive. So if you see a morphology that's indicative of AFB, for example, if there's a lot of bunching, so like tuberculosis on the gram stain can show like a bundling or bunching. So basically when you're reading a sputum, for example, you may think you see AFB, and out of a precaution you'll do the Kinyoun stain to work that up off of the gram stain. So that's one time when you would do the Kinyoun stain. Another time is directly from a culture. You have growth on a culture, and it looks like the colony. It looks like the colony is AFB. Then you could do a gram stain on that to confirm or deny your suspicion as well. And then primarily, the main time you do it is that when you get an AFB culture, that's going to have an AFB smear with it as well. But that smear is not the Kinyoun smear. It's actually a fluorescent smear that you do called Oromine O stain. And the reason you do that stain instead of the Kinyoun stain is just because it's easier to read and interpret. Because it's fluorescent, you can kind of just scan the slides very quickly. And whenever you see a green rod on the Oromine O stain, that would be a suspected AFB positive organism. And so when you see that, you would then do the Kinyoun stain as a confirmatory stain. And what you have here is a Kinyoun positive stain. So remember, that's going to be pink or red, and the same organism would have stained gram positive as well, or purple. And one thing to keep in mind is, remember, with your Nocardia species or with other partially acid fast organisms, they're going to stain negative actually for the Kinyoun stain, but positive for the modified Kinyoun stain. And the modified Kinyoun stain just has a slightly weaker acid alcohol decolourizer, and so that's why it will stain positive for that stain. So yeah, that's just one thing to keep in mind there, and hope you know more now about the Kinyoun stain and AFB in general as well, between Mycobacterium and Mycolicibacterium. And thanks for watching, and I'll see you next time.